Good day, sir. By the way, I am Junel Fortune Miguelin Jr. And the topic that we are going to discuss to you, sir, it's all about what are the risks and challenges of gene therapy and stem cell research. Gene therapy involves altering the genes inside your body cells in an effort to treat or stop diseases. Gene therapy has some potential risk. A gene cannot easily be inserted directly into your body cells. Rather, it usually has to be delivered using a carrier called a vector. The most common gene therapy vectors are viruses because they recognize certain cells and carry genetic materials into the cell's genes. Researchers remove the original diseases-causing genes from the viruses, replacing them with the genes needed to stop diseases. Further discussion about risk, sir, will be discussed by Ms. Juliet Piyonko.
Now, this technique presents the following risks. We have unwanted immune system reaction. It means that your body's immune system may see the newly introduced viruses as intruders and attack them. This may cause inflammation and, in severe cases, organ failure. Another one is that targeting the wrong cells. Why? Because viruses can affect more than one type of cells. It's possible that the altered viruses may infect additional cells, not just the targeted cells containing the tainted genes.
What can you expect? Currently, the only way for you to receive gene therapy is to participate in a clinical trial. Clinical trials are research studies that help doctors determine whether a gene therapy approach is safe for people. They also help doctors understand the effects of gene therapy on the body. Your specific procedure will depend on the disease you have and the type of gene therapy being used. For example, in one type of gene therapy, you may have blood drawn or you may need bone marrow removed from your hip bone with a large needle. Then, in a lab, cells from the blood or bone marrow are exposed to a virus or another type of vector that contains the desired genetic material. Once the vector is entered the cells in the lab, those cells are injected back into your body, into a vein, or into a tissue, where your cells take up the vector along with altered genes. Viruses aren't the only vectors that can be used to carry altered genes into your body cells. Other vectors being studied in the clinical trials include stem cells and liposomes. Stem cells are cells from which all other cells in your body are created. For gene therapy, stem cells can be trained in a lab to become cells that can help fight disease. Liposomes, these fatty particles, have the ability to carry the new therapeutic genes to the target cells and pass the genes into your cell's DNA. The results of gene therapy. The possibilities of gene therapy hold much promise. Clinical trials of gene therapy in people have shown some success in treating certain diseases, such as severe combined immune deficiency, hemophilia, blindness caused by retinitis pigmentosa, and leukemia. But several significant barriers stand in the way of gene therapy becoming a reliable form of treatment, including finding a reliable way to get genetic materials into cells, targeting the correct cells, reducing the risk of side effects, and gene therapy continues to be a very important and active area of research aimed at developing new.
The challenges of gene therapy field. It has evolving for decades despite the best effort of researchers around the world. However, gene therapy has seen only limited success. Why? Gene therapy possesses one of the greatest technical challenges in modern medicine. It is very hard to introduce new genes into cells of the body and keep them working. And there are financial concerns. Can a company profit from developing of gene therapy to treat a rare disorder? If not, who will develop and pay for these life-saving treatments?
Let's look at some of the main challenges in gene therapy. Gene delivery and activation. For some disorders, gene therapy will work only if we can deliver a normal gene to a large number of cells, say several million in a tissue. next main